Hi, my name is Dr. Swarnava Roy, and I will be talking about meiosis in this lecture. At the bottom, you have all the keywords I will cover today. That includes the different stages of meiosis, mitosis, the difference between chromosomes and chromatids, and the concepts of haploid and diploid, and the meiotic and the mitotic cell cycle, homologous chromosomes, tetrad formation, and crossing over, and the significance of crossing over. Now, human beings have 23 pairs of chromosomes, which is equal to 46 chromosomes. So each somatic cell, or a cell from the body, it has a total of 46 chromosomes, and these chromosomes are in pairs. So that is the entire set. So out of the pair, one chromosome comes from the mother, which is the maternal chromosome, and one from the father, which is the paternal chromosome. Now, this set is taken from a male, and that's why you have the X, Y sex chromosomes, and the other 22 pairs are the autosomes. So, as you can see that the chromosomes are in pairs, which are known as the homologous autosome pair. And out of the pair, each chromosome is again made up of two chromatids. So you can see they are called the sister chromatids, and these chromatids are joined at the centromere. So 23 pairs of chromosome equals to total of 46 chromosomes, and each chromosome is again made up of two chromatids. So 46 chromosomes equal to 92 chromatids. Now, the bottom picture shows the difference between diploid, or 2n, and haploid, or n. As you can see that on the left side, the diploid cell has, as I said, 23 pairs of chromosomes, the red or the pink and the blue. So each of those chromosomes coming from either the mother or the father. That's homologous chromosome. And on the left side, on the right side, I'm sorry, we have the haploid cells, which is N, and you see that the blue chromosomes are not there, just the pink set, which represents the 23 chromosomes of the haploid set. So now we have another example of the haploid and the diploid there, but I'll just go quickly because now we understand the concept of haploid and diploid. Now this is a life cycle of haploid and diploid, and you can see that how the two parents, they have their gonads, the testis in the males and the ovary in the females, and these gonads, they make gametes, and the gametes they represent the haploid part of the life cycle, which is N, or 23 chromosomes. The egg and the sperm, they fertilize, they fuse, and they form the zygote. The zygote is 2N, which is 46 chromosomes, which is diploid, and this zygote then divides and divides and forms the multicellular human being, where the somatic cells are 46 chromosomes, which is 2N. And again, the parents, they actually produce gametes, and the gametes mate, and so the cycle of haploid and the diploid transition, it goes on. So next we compare the cell cycle of a mitotic cell, or a cell undergoing mitosis, and the cell cycle of a cell undergoing meiosis, and compare the differences between the two. And you can see that the interphase part, which is the blue part, which comprises of the G1S and the G2, is similar between a cell in mitotic division and a cell in meiosis division. And so the only difference is the division phase. You see that's the red box. As you can see that in case of the mitosis, the mother cell, which is diploid, it divides into two daughter cells having the same number of chromosome. In other words, the chromosome number is maintained in case of the mitosis as its parent. The meiosis, however, the division phase is different from the mitotic division phase, where you see that the green, the division, the green bar, it divides two parts of the meiosis 1 and the meiosis 2. The meiosis 1 is known as the reduction division, where the chromosome number between the mother and the daughter cells is reduced from the 2n to the n. And the meiosis 2 is an equal division, where the chromosome number between the mother cell and the daughter cells they remain the same from n to n. So this is an overview of the entire mitosis on the left side and the meiosis on the right side. And it shows you the difference between the two types of divisions, and you can see that the meiosis has the first stage, the first set of divisions now, which is known as the meiosis 1, which comprises of prophase 1, metaphase 1, anaphase 1, and telophase 1. And this is the part that is different from mitosis. So the meiosis division has an extra step, which is meiosis 1 or reduction division, where the 2n diploid chromosome number of the germ cell is reduced to n, or the haploid set, in the daughter cells. Now, in the lower red box, which compares the mitosis with the meiosis 2, the meiosis 2 is exactly same as the mitosis. It again has the steps like prophase 2, metaphase 2, anaphase 2, telophase 2, and you can see that how the chromosome number between the starting cell and the end daughter cells remains the same. So the meiosis 2 is similar to mitosis, and the different points. The first point, the chromosome number remains the same between the mother and the daughter cells. Number 2, all the steps are similar between the two processes, and in meiosis 2, no crossing over step occurs, unlike meiosis 1. Now we will discuss about crossing over and the significance of crossing over in the following slides. So here we have a cell, which is, it's a germ cell. That means it's diploid, and this cell is about to undergo meiotic division. Now, for simplicity, we have taken just 3 pairs or 6 chromosomes. And when I say pair, so the chromosomes are short and condensed just before the start of prophase 1. For simplicity, as I said, only 3 pairs of chromosomes are considered, and this is the germ cell from which haploid daughter cells will emerge. Now, you can see that each pair of purple and blue, it constitute the homologous chromosome. So you can see that those red arrows from the purple to the blue, they are the homologous chromosomes, one from the mother, one from the father. Okay, and so now those orange cylinders are the centrioles, and before the beginning of the prophase 1, these centrioles, they move towards opposite poles. Okay, and the centrioles, they have microtubules emerging from them, and these microtubules form the spindle fibers, and it is on these spindle fibers the chromosomes, they will come and align. So we will talk about that in the later stages. So this is the prophase, beginning of the prophase. We have we see that the homologous chromosomes, so these are the sister chromatids, and those are the homologous chromosomes. They align with each other, and they form what is known as the tetrad. So tetrad formation is very important for meiosis, because tetrad will result in crossing over. That means one chromosome from each of one chromatid, to be more specific, from each of the non-sister chromatids. Remember, they will exchange parts with each other. So we'll talk about that in details. Here is crossing over, and you can see that how the pink chromosome and the blue chromosome, the non-sister chromatids, okay, they are actually exchanging parts or segments with each other. So you see that the red box, how the blue chromatid is lying on top of the pink non-sister chromatid, and how in the second picture you see that how the parts have been exchanged, and how the pink chromosome now has a blue leg, as I can say, and the same vice versa, the blue chromosome has the pink portion. So what it means is all the genes in the blue lower part has been shifted to the pink chromosome. It's called gene shuffling, and this is one of the biggest reasons why we are unique. Okay, that's why we don't exactly look like our parents. We're not clones of our parents. At the same time, all the siblings are also different from each other now. It's because of this gene shuffling. It happens, and it gives different permutation and combination. So that's the significance of crossing over. What's relevant to us for this lecture is through crossing over, segments of homologous chromosomes are interchanged and hence provide origin of new characters and genetic variations. And that's why our species, it keeps evolving. And if there would have been no genetic variation, no crossing over, then we would cease to exist. So this is the most important mechanism or step, which is preserving our species, you can say. Now, the crossing over has helped in establishing the concept of linear arrangement of genes. So we know that the genes are arranged in a linear fashion, and that's crossing over. Scientists have known that by crossing over. And also the frequency of crossing over. The frequency of crossing over helps in mapping of chromosomes. Okay, now that is determining the location of genes in the chromosomes. That's all we can do it by studying crossing over. Now, this is the pro-metaphase 1, and one important concept we need to know is the chromosomes now, they get attached to the spindle fiber, and they start migrating towards the middle metaphase plate that will come right at the equator of the cell. But before that, we need to know that meiosis versus mitosis, and we need to know the spindle fiber location. Now, in meiosis, the spindle fiber, it passes right between the two homologous chromosomes. You see that arrow? So the yellow spindle fiber, it just is present right between the two chromosomes, the two homologous chromosomes. While in the mitosis, the spindle fiber is attached. So each chromatid, each sister chromatid, is attached to the kinetochore. You see those green balls where each of those purple sister chromatids are attached. So what happens? Due to this attachment, we have two things happening. Now, in case of meiosis, we see that how when the spindle fiber breaks, you see in meiosis, the whole chromosome, that means comprising of two chromatids, migrates to opposite poles. You see, that is the reason why we have reduction of the chromosome number, because the entire chromosome is moving towards opposite poles. But so results in the entire chromosome moving towards opposite poles after the spindle fiber breaks during the later phase of the meiosis. You will see that in the Anaphase, how that spindle fiber will break and will result in moving of the entire chromosome. Unlike that, in mitosis, we have the break happens between the two sister chromatids. So now we have one chromosome is split up, and each chromatid is now moving towards each pole, and then they are then in the S phase of the cell cycle. That's those chromatids, they duplicate, they replicate, actually, as the term, into another chromatid. So that's why in mitosis the diploid state is maintained between the mother and the daughter cell, but in meiosis the reduction division happens, the chromosome number reduces. That's the main reason, because of the positioning of the chromosomes on the fibers during the pro-metaphase. So in mitosis, however, the individual chromatids move towards opposite poles, and after the spindle fiber breaks during the later phases. So that's the red box showing how they're moving. So in metaphase 1, the chromosomes, the homologous chromosomes, which are in the tetrad and which have just finished crossing over, and you can see that how they have the purple chromosome has some blue part inside, right? And the blue chromosomes have the purple. So the shuffling has taken place, and now they are arranged on the metaphase plate. And what happens? So that's the metaphase plate, and then in the anaphase, the break happens in the spindle fiber. Now, as I mentioned before, the entire chromosome, okay? So one chromosome for the mother and one from the father, moving towards opposite poles. And you see this whole thing, which side the blue will move, which side the purple, that's totally random. So that causes another level of genetic variation. So your brother might have, blue, purple, and blue moving towards one pole, and you might have purple, purple, purple moving towards one pole. You see, so that again causes another level of genetic variation. And again, this explaining why siblings they look different, and why we are not clones of our parents, and siblings are not clones. So that's another level of permutation combination happening. So in the telophase, the chromosomes have moved further towards the pole, and the most important thing, you see that the cell starts dividing, the invagination or the cleavage has started. And in the later part of the telophase, you see that the cleavage, it deepens further, and you see that we can see now that the nuclear membrane, starts to appear, the faint nuclear membrane. So they start to appear. You see that the red circle, which the nuclear membrane formation is complete in the cytokinesis. So here in the cytokinesis, the cleavage or the separation of the two cells, it becomes more prominent, and you can see that the end of the cytokinesis, you see that the two daughter cells are completely separated. The nuclear membrane is now conspicuous. You see a completely well-defined nucleus is formed. And at the later part of the cytokinesis, you see that the chromosomes, which were condensed all the time and short, they are now getting longer and thinner. So at this point, if you look at the cell under a microscope, it will be very difficult for you to visualize the chromosomes, because now they are unwinding and they are decondensed. That's the word. It's very hard to see now as the cell enters the later part of the cytokinesis. So now we move on to the prophase 2. Okay, remember the meiosis has meiosis 1 and meiosis 2. So this is the meiosis 2 is beginning. So in the prophase 2, what happens is the condensation of the chromosome starts happening again. Remember, the chromosome was decondensed. They were thin thread-like, but now they're again getting thicker, and the nuclear envelope dissolves. Okay, so there's no nucleus, and the chromosomes start moving towards the metaphase plate. You see that the centrioles, they move again towards the opposite poles, and the mitotic spindle forms, and the chromosomes start moving towards the plate. You can see there. The metaphase 2 happens, and we have the metaphase plate formation, and the chromosomes, they move right to the metaphase plate. And then comes the anaphase 2, where the spindle fibers, they split in the middle. And now here, as I said, the meiosis 2 is just like mitosis. And so you see that each chromatid is now moving towards the opposite pole, unlike the whole chromosome moving. And that's why between the starting cell, or we can't say a mother cell here, but between the starting cell and the end cell, the chromosome number is maintained. So anaphase 2, you see that now the cleavage starts happening in the later part of the anaphase 2. The chromatids are deep inside the opposite poles. In telophase, the invagination, it becomes deeper, and the cells are now almost divided. And you see that how 4 daughter cells are emerging, and again the nuclear membrane starts forming at the telophase. And this nuclear membrane now becomes complete at the cytokinesis 2, and also we see that the chromosomes are again decondensed, and they are thread-like. And again, at this point, if you put a microscope, it becomes difficult for you to visualize the chromosomes. Unlike when the cell division starts, when the cells are condensed, it's much easier to see the chromosomes. So this is the overview of the meiosis and the mitosis. And you can see that the meiosis has meiosis 1 and meiosis 2, which is different from the mitosis. And the meiosis 2 is similar to mitosis, where the chromosome number is maintained, while in meiosis 1, there is reduction division, the chromosome number reduces. So finally, we come to an overview. So as I explained, that the parents, they have gonads, and the parent has somatic cells, which are 2n diploid, that is 46 chromosomes, and the gonads, they make the gametes, and the gametes are either the sperm from the male or the egg from the female, and these are haploid. They are produced by the process of meiosis, and they have the 23 chromosomes, and then there is fertilization, and a zygote is formed, and then develops into a complete child, and the child is 2n, and again the process happens. So these are actual pictures of meiosis 1 taken from the microscope, and you can see that the different phases are mentioned. You can see the metaphase, how the chromosomes are aligning in the metaphase plate, and then the anaphase and the telophase. And then we have the meiosis 2, again, actual pictures taken directly from the microscope. And then you see how the prophase 2 forming, the metaphase 2, and the anaphase, how they're moving towards the opposite poles. And then the telophase, the cleavage starts, and by the end of the cytokinesis 2, we have 4 daughter cells, which are actually haploid. Now, this is another picture of the different stages or the steps of the meiosis, and you see that we can look at the alphabets, A to I, and as you see, A is metaphase 1, anaphase 1, and then C, telophase 1, and you can see the D is the cytokinesis, where the cells are divided, and the reduction division has taken place. And then we have the meiosis 2, where you have the metaphase, where all the chromosomes are aligning on the metaphase plate. And then we have the anaphase 2, where again they are moving towards the opposite poles, which is F. The G is the telophase 2, where the cleavage will start, and H, cytokinesis, where the cleavage is complete, and then I, it results in 4 daughter cells, haploid gametes. So, so that's the end of the lecture, and I hope I've been able to make you understand the different steps of meiosis and the various concepts of diploid, haploid, and chromosome chromatid. You can visit my website, which is www.swarnavaroyphd.com. I provide assignment help. I also conduct regular lecture over Skype, and I can guarantee you good grades, and I can that will help you succeed in your career. Thank you, and bye-bye.